Thank you for the invitation. My name is Winnie Svendsen, and I'm a professor at DTU Bioengineering. And together with Maria Dimaki, a senior researcher at DTU Bioengineering, we will present a short tutorial on biosensor introductions from fabrication to application. We are from the Technical University of Denmark, working in the research group Nanobio Integrated Systems. The vision of NABIS is to integrate micro and nanotechnology in lab-on-chip systems for biological analysis, including medical diagnosis, treatment monitoring, and environmental monitoring. We work with a lot of different sensor platforms, from silicon nanowires, impedance spectroscopy, to electrochemical sensor platforms. We also include sensor platforms like lateral flow assays and even droplet microfluidics. Today, we will touch upon some of these sensor systems in our talk. So, what are sensors? Sensors are everywhere in our daily life. They're all around us. They're all different types of sensors. We encounter temperature sensors, motion sensors, pressure sensors. And actually, one of the things that we actually use every day probably has the most sensors of anything. That is our car. In our car, we have sensors everywhere. We have infrared sensors, radar sensors, microphones, ultrasonic sensors, fuel sensors, camera sensors. There are tons of sensors in the car that we drive in every day. Also, sensors are moving the way into our personal health and body. So we have a lot of sensors that we can integrate into our own systems. We have the watts, where we can monitor our heart rate, our sleep, the blood pressure sensors. We have insulin pumps that you can work with every day. So sensors are used everywhere around us and are really important for our society. So what are sensors and how do we use them? Well, first of all, if you want to use a sensor, you need to have some kind of scale, some kind of thing that you can measure. So let's look at the temperature sensors and the temperature scale. How did you make it possible to measure temperature? Well, actually, one of the first sensors that exist, and the scale was invented by the first scale to be measured was invented by Robert Hooke, where he assigned a 0 point for the freezing temperature of water. Later on, the scale was expanded by Ole Rømer, where he added another point of the temperature scale, which was the temperature of boiling water. Ole Rømer put the boiling water temperature to 60 points. Later on, Fahrenheit introduced his very complicated temperature scale based on the 0 point as the human temperature, 37 degrees. And later the Celsius scale came, which was an improvement or a change of the Rømer scale, where the boiling point of water was placed to 100%, 100 degrees Celsius, it was called at that point. So we had a scale from 0 degrees Celsius at the freezing of ice, or freezing of water, and then 100 degrees Celsius at boiling of water. So this was a temperature scale. Later on, when we could measure even lower temperatures, the Kelvin scale came, which was the absolute zero started point as a starting point. These temperature scales are still used today, the Fahrenheit, the Celsius, and the Kelvin scales, and you can convert in between the three temperature scales. So now we can measure, we have something that we can relate to as the temperature. So how do we measure this temperature? Well, there's a lot of different sensors from history. The first temperature sensor was made by Galileo and Santorio, Hooke and Rømer, all of them together more or less at the same time. The Galileo temperature sensor was a very simple system consisting of a glass ball placed in some liquid, and when the temperature changed, the liquid raised up in the tin cylinder, and then you could measure the temperature relation with using a ruler and relate that to the boiling point and the freezing point of water. This temperature sensor was actually used in medicine at a very early stage. It was used to measure the temperature of the body by placing the mouth over this ball, and then you could see how far liquid weighs, and then you had an indication of the temperature of the personal temperature, which of course was an indication of your health, whether you were sick and healthy. So it's already at this point in time, this sensor had an impact on human health. Now, later on, Römer and Fahrenheit developed a more simple sensor, a temperature sensor, and this has been used, actually, it's still used today, where the first sensor was based on alcohol, where it heated up a color liquid and moved up, and you measure the temperature. And then the mercury sensor, which was used recently when you found out mercury was poisonous. So a question to consider, is the thermometer a biosensor? It did have a huge impact on human health. Start. So biosensor, what are biosensors? The definition of a biosensor in general terms is that a sensor which detects biological species like cells, viruses, bacteria, protein complexes like antibodies, and even chemical compounds important for biology is a biosensor. There's also a more scientific definition from Institute of Union of Pure and Applied Chemistry, which define a biosensor as an integrated receptor transducer device which are able to provide selective, quantitative, or semi-quantitative analytical information using a biological recognition element. So, was a temperature sensor a biosensor? No, it's not. By this definition, it's not a biosensor, even though it has an impact on our physiology and our human health. So it's a physical sensor like a lot of the other sensors that I have mentioned so far, pressure sensors, displacement sensors, electromagnetic sensors. All these are physical sensors, but a lot of them can be turned into biosensors. So let's talk about biosensors. The essential part of a biosensor can be described by 2 or 3 essential elements in spatial proximity. A biological recognition element able to interact specifically with a target analyte. A transducer able to convert the recognition element into a measurable signal. And finally, a readout is often needed for humans to identify the measured signal. So we will look into these 3 different components to make a sensor into a biosensor. So schematically, we can represent it like this. You have an analyte, what you want to measure. Then you have a biorecognition element. You have some kind of interface to couple the recognition element together with the transducer. Maybe you need an amplifier, and then you need an output. So look at these components. So the analyte and biorecognition element. The analyte is the sample containing the specific biological species we want to detect. For instance, sugar in the blood, glucose in the blood. The glucose sensor is the biosensor. It's also used for describing the specific biological species to detect. The biological recognition element is the tag that ensures that we can only detect the biological species in questions in the analyte and nothing else. So we don't want to detect anything else than our analyte in question. The biological recognition element, let's look at that. It has some needs to be able to ensure selectivity, must be inert to other compounds in the sample. It needs to ensure stability of the sensor, so have a slow degradation, and it needs to be sensitive, so a strong bonding or affinity with the target analyte is also a good thing to have. The recognition element interaction types are several. Here I have listed a few. Now we have the DNA-RNA or DNA-DNA interaction, where the single-stranded DNA can bind or recognize the other DNA strand. We have the receptor-ligand interaction, where a specific receptor will detect a ligand. We have protein or molecules, molecular interactions, where you have a specific protein that is a receptor of some kind to detect the molecules in question. Then we have two that we will talk about today, the antibody-antigen interaction, where one of these can be the biorecognition element and the other can be the analyte. This is very common used in central labs for ELISA today. And then we have the enzyme substrate reaction, where you have an enzyme that is altering your analyte so that you can measure it. So if you look at the enzymatic reactions, it's illustrated here how that works. So in the first step, you have some kind of substrate entering the active site of the enzyme. So here you have the substrate and here's the active site. The enzyme changes shape slightly as the substrate binds. Then a chemical reaction occurs and you will end up with two products. And these two products, or the products, it does not need to be two, this has a unique fingerprint of your actual substrate or your analyte that you wanted to detect in the first time. And this is what we then want to detect in, for instance, an electrochemical sensor. The immunological or antibody-antigen sensor works slightly different. Here is the analyte, the biological recognition element is an antibody, or it can be an antigen. The antibodies are what is produced by mammalian immunological system as a response to an intrusion by a foreign molecule, also known as the antigen. So the antigen is often the foreign molecule. So the antigen is a natural, or it can be a synthetic substrate recognized by the immune system, able to introduce an immune response or to react with a receptor or the antibody. So here we have a system where you can use it as an analyte or as a biological receptor system. It's very strong because these are created in the human body as biomarkers. So how do we functionalize the sensor? How do we give this sensor a function so it can convert into a biosensor? We do this by attaching the biological biorecognition element to the transducer surface. And we do this by this interface layer. Now the challenge is how to attach a biological organic molecule to an inorganic surface, gold, silicon, silicon oxide, which are often the material of our sensors, and then at specific locations. The solution is, of course, glue. And what is the glue between the biorecognition element and the transducer of the nanomolecule? And this is, of course, surface chemistry. So we want to look into surface chemistry to make our physical sensor into a biosensor by this intermediate layer. Now, what kind of bindings do we have? We have different types of bindings, biological bindings. We have absorption, physical entrapment, covalent coupling on the surface of the transducer to the biorecognition element, covalent coupling on an intermediate polymer layer maybe, and non-covalent coupling. We would, of course, prefer covalent coupling because that provides the strongest bond and a stronger and more robust and more reliable sensor. What is it that we want to bind to? I mean, if you look at biomolecules, they often have very specific reactive groups, such as amino groups, carboxylic groups, which are placed on specific location, for instance, on the antibody here. We would like to be able to attach these reactive groups to the surface of our sensors. And how do we do that? We do that by modifying our surface of our sensors with specific chemical reactions. For instance, with metals like gold, we are very lucky that we have a tile group that contains sulfur hydride group. It has a very strong odor, but it attached to gold and also a little bit to silver. So if we can attach this tile group onto the surface of gold, then it will react with the reactive groups of the biomolecule. So you have to make these complicated chemical layers on the surface of your gold to actually bind your bioreceptor. This is really good for gold, but what about silicon? So let's look at the silicon surface. When working with silicon, you have the possibility of also working with silicon oxide, or you can remove the oxide layer so you have a hydrogen terminated surfaces. So what are the possibilities and what to choose? The strategies for silicon functionalization can be to use the surface as oxide or without the oxide. And there's two methods that I will go through, or I will go through some of these methods. You can produce oxide, a silicon oxide surface by Cytometry. You kind of get a type of binding that is covalent, which is what we would like, and then you have this monolayer, a thin layer on the surface. If you want to remove the oxides of the surface, you can do hydrosolinization. This is also a covalent bending and monolayer. So they are two very good methods to make an inorganic surface into an organic surface. So let's look at silicization. So you have a slab of silicon, and then you do hydrosolization using what is called a Piranha treatment. When you've done that, you have a silicon surface with the OH dangling bonds on the surface. These OH dangling bonds can be used to bind biomolecules, like a molecule that is used very often is called Aptus, and I'm not going to try to pronounce its real name, but let's just call it Aptus. And then Aptus has these three reactive ends and a silicon here. What happens is that Aptus will bind to the silicon surface via these OH reaction and create this monolayer of an organic material which has amine dangling bonds in the end. And this is good because if you remember, our analyte often have these reactive groups, which is also amine groups. So what we need to figure out is how to combine our biorecognition element, which has one amine groups, to the surface of the silicon, which has another set of amine groups. If we can do that, we actually have a good adhesive biorecognition element on our surface. And we can do that with gluing this together by a molecule called glutaraldehyde. And glutaraldehyde has a biofunctional layer, so it has the same reaction at two sides. So if you have the sensors with amine groups at one side and the protein of our biorecognition layer at the other side, also with amine group, this glutaraldehyde will link the two together. So now we have made a monolayer of organic material on the surface of our sensor, of our silicon sensor in this case, where we have a protein of a kind as a biorecognition element. So we have achieved the most important thing for making our sensor into a biosensor by attaching a recognition element on the surface of our sensor. The next step is hydrosolinization. It's a bit more tricky because it's very hard to move to keep silicon without oxides on the surface. It has to be done under very controlled environment. But the way to do it is to have the silicon wafer, then you remove the native oxide by hydrofluoric etch down, and then you can react with alkanes or alkynes on the surface, making a organic surface on the surface of the silicon, and then you have to treat it with UV light or with heat at room temperature to get in an oxygen-free conditions. That actually results in monolayer where you don't have any more oxidization of the base silicon layer. And then you have a stepping stone to attach your biorecognition layer exactly in the same manner as we did with the glutaraldehyde. When you have made the biorecognition layer on the surface, then you have to look at what kind of transducer you want to use. And the transducers are the actually sensing element of your biosensor.
So you have made the surface of this transducer now organic by using surface chemistry, and then you want to do the actual measurements. So the transducing element transfer the event of an analyte being bound on the surface by the biorecognition element into a measurable physical quantity. And what can that be? It can be everything. It can be an optical transducer, where we use fluorescence or color change binding to the biorecognition element. We can have an electrical signal, where we can measure current when the binding occurs, or mechanical signal, where, for instance, we get a bending, a surface stress change in the surface of the sender when a reaction occurs. Now, the transduction X element itself is usable, unable to distinguish between the target analyte and a molecule with similar physical properties. It can only tell you something is attached to the sensor, but not what it is. So that is why the biorecognition element is what ensures that the something is an actually target analyte. And this is what makes our normal sensor or our transduction element into a biosensor when this is happening. We can turn our sensors or transducers as label-free labels versus label-free. When you have labels, you rely on the text of a specific label. It could be a fluorescent tag attached to the biomolecule of our interest. Now, the label-free relies on the text of the direct measurements. When the reaction that occurs is, for instance, a change in electrical conduction of, for instance, a nanowire, or current due to oxidation of a molecule, electrochemical, and then you measure the current in a direct relationship to the analyte of interest. This is, of course, generally preferred because you're probing your analyte of interest directly and not relying on an intermediate reaction. So colorimetric assays are often used as a labeled transduction element is an optical transductions. You can use chemoresponsive dyes, and when the dyes react with your analyte of interest, you get color change. And then you can detect it either by the eye or by an optical measurement or by a camera. This is a method, for instance, that can be used to detect or volatiles for instance, diagnostics of malaria. Other ways of using colorimetric assays is using metallic nanoparticles like gold, and you bind them with an antibody in a solution, and then when the antigen reacts with the antibody, you get this aggregation, and then you get a color change with optical spectroscopy. And this is also a very precise method to measure antigen bindings, but you don't have the specificity of looking at a specific binding event individually, which you can do with very sensitive nanosensors, which we'll discuss later. But before we do that, I will just give you another example from one of the very, very early biosensor systems for kind of point of care use in the old days. So let's look at the pregnancy sensor system and its involvement over the years. Now, the first pregnancy sensor was actually invented in Egypt 1350 before Christ. It was used as a barley sensor. You will have a bag of barley or a bag of wheat, and then when women would urinate onto this, the barley would either not grow very rapidly or grow very rapidly. And if it grew very rapidly, then it indicated that the woman was pregnant. And this was due to the hormones in the woman's urine that was fertilizing the barley. And in that way, you give you an indication of the pregnancy of the woman. So very early and very primitive biosensors. But it's actually the same principle that our modern days biosensors are based on. So the evolution of the pregnancy testing is quite interesting, actually, because you had the old Egyptians that did it in this very humane way of just peeing on barley. But in our recent time, the started doing, when you started investigating biochemistry, you actually did this test by injecting the woman's urine into mice, later into rabbits, and then you killed the mice and the rabbit to see if it had ovulated, if it had started being pregnant. Later on in the 30s, you did this on frogs, and you didn't have to kill the frog because then it would start laying eggs if the urine was from a pregnant woman. So this was, of course, done at the doctor's office. I mean, you don't kill mice and rabbits at home yourself, so it's not kind of a personal test like it was in Egypt. But in the 60s, things changed. You started doing a blood analysis, and you start working with antibodies and antigens testing methods. And you had developed an antibody system towards HCG, our human pregnancy hormone. And then you did blood tests where you tested the blood, and you then made ELISA to see if you had HCG in your blood, and then you could tell if you were pregnant. Later on in the 70s, a shift changed where you could go from blood to urine, which made a test easier. But it wasn't until in the 80s that the actually home test or the point of care test in the dipstick was used, came to the market. And how does the dipstick or this standard pregnancy test that are used today work? It's what's called a lateral flow assay. So what the essential part of a lateral flow assay is the paper. The paper is a strip of paper where you put these fluorescent dyes on the surface of the paper. And that paper is then dipped into the liquid where the analyte of interest is. And then the paper will change color if that analyte is on the surface. And that paper was in the 80s integrated into a stick for only HCG. So when you peed on the stick, and then you will get these two lines whether you're pregnant or not. How does that work? It's actually it's illustrated here. So the architecture of lateral flow assays are that you have, that you have an analyte with your HCG on your drop of the liquid here, urine with HCG in it. Then you have the analyte conjugated to the gold nanoparticle that we talked about before. Then the antibody will flow together with the gold nanoparticles down the strip of paper, and then you have these antibody placed, the biorecognition element placed on the surface of the paper. Then if you have the antibodies in your for HCG in your analyte, in your blood, in your urine, then they will bind on the surface of the strip, and then you get one line, and then you have these free antibodies in that will also bind if it's a control line, just showing that the test actually works. And then you get two lines, and then to indicate if you have a pregnancy. And the lateral flow assays is the base for a lot of the point of care tests and one of the most simple biosensors that exists today. Another biosensor that is very interesting to look at is the electrochemical biosensor. This is one of the other most common biosensor that exists. It's used for monitoring of the blood sugar, the diabetes monitoring, the blood sugars, the glucose in the blood. And it's an enzymatic reaction that we talked about earlier on. The analyte is glucose placed in blood sample. The biorecognition element is an enzyme called the glucose oxidase enzyme that reacts with the glucose in the blood and releases an electron, and that electron is transduced, is recorded by the transducer, and then a current is read by some kind of equipment, and you get a signal which is directly related to the amount of glucose in the blood. This can be done in the lab in these very large equipment, but of course, this was developed as a very small device in the 70s and changed the life of people with diabetes before in the 70s, before the 1970s, diabetes was actually a disease that you died from. Where after the glucometer was invented, people can live a very good life, and a long life, having diabetes. This was, say that this is definitely a biosensor that had changed the humanity's life expectancy. So these are the most common commercial biosensors are based on electrochemical biosensors. So today you can get them very, very small and even a small patch on your arm.
We will now continue with another very interesting biosensor, namely the silicon nanowire field effect transistor. This is a system based on electrical transduction, where a charged molecule in the vicinity of the nanowire will affect the charge carriers inside the semiconducting channel, and a change in conductance will be measured. This is a sensor that has the potential to be very, very sensitive, and indeed this is shown here in this very nice work by Patolsky in 2004, where even a single virus particle that comes in the vicinity of a nanowire will change the conductance enough so that it can be measured. And here they show that one particle comes there, close, and the change in conduction, they see the particle moves away, and then the second particle comes and they see exactly the same change. Now, the concept of the biological field effect transistor comes actually from the original transistors, the MOSFETs, and also the ESFETs, so ion-sensitive field effect transistors. Now, in the original transistor, we have a p-type silicon and highly doped contacts, and if we apply the correct voltage on the gate, then a semiconducting channel will open from the source to the drain. If you have the gate insulator in direct contact with the solution, and then you functionalize this surface of the gate, then you can detect specific species in the electrolyte with this system. In the silicon nanowire-based BioFET, the nanowire is always there, so the semiconducting channel is there always, and it's always conducting all the time. But how much it is conducting depends on the amount of biomolecules that act as a gate on top of nanowires. And you can think of it as a water pipe with a valve. So how much water will go from one side of the pipe to the other depends on how open the valve is. To further understand what affects the function of a silicon nanowire field effect transistor, we use a theoretical model where we model the nanowire as a cylinder that has a thin layer of oxide and then is placed inside an electrolyte. And then the biomolecules attaching to its surface are modeled as charges that are situated a certain distance away from the surface of the nanowire. We define the sensitivity of the nanowire as a change in conductance divided by the base conductance. Now, if you solve all the physics and all the differential equations, then you end up with the expression like this for the sensitivity, which shows that the sensitivity is of course dependent on some physical parameters from the nanowire, so the radius and the charge carrier density, and then by two terms, a term that describes the intrinsic screening that this system experiences, a nanowire experience, just because it is inside an electrolyte and because it has a certain charge carrier density. And then another term over here, this sum, that describes the additional screening that comes from the charges of the analyte. And this depends from the distance of the charges from the nanowire surface. So what does affect the sensitivity of the nanowire? Well, it's all actually about these charges. Sensitivity is limited by screening of the electric field inside and outside the wire. If we focus on the outside of the wire, then this is an electrolyte. That means there is free ions here, and depending on the concentration of the electrolyte, then there will be a double layer forming on the surface of the nanowire. Now there is this characteristic length that we can calculate, which is inversely proportional to the concentration of the electrolyte, and basically this is a length that the physical meaning of this length means that any charged species that are situated further away than the Debye length from the nanowire surface are basically invisible to the nanowire and will not have an effect on the wire conductivity. Typical values of this length are 1 to 10 nanometers, so you can see that we don't actually have a lot of playroom. And this is a very nice paper that illustrates exactly the importance of the Debye length. So here we have a nanowire that is functionalized with biotin, and then a streptavidin has been added on top and binding to the biotin. And originally this nanowire is placed in a low concentration electrolyte. This is shown here, illustrated here by the orange line. And when this happens, so when we attach the streptavidin, we see that the conductance changes to a certain level. If we now exchange the electrolyte to something that is has a bit more concentration, which is now indicated by the blue line, so high concentration, the lower the Debye length. So now we are losing some of the charges of the streptavidin, and you can see that indeed the change in conductance is now smaller. Notice that we haven't removed any streptavidin. We have exactly the same amount of streptavidin, but because of this screening of the charges, we see simply a less change in current through the nanowire. If we now further increase the concentration of the electrolyte, then we actually end up losing the entire signal. And if we now exchange the buffer back to the original low concentration buffer, then we get again our signal. If we now remove the streptavidin by just cleaving it away, we can see that we go also to the original background signal. Similarly, we also have screening inside the wire because, of course, the nanowire contains these charges. And there is also a screening length here called the depletion length that basically means that only the charges situated this distance away from the surface of the nanowire will be affected by the gate, so by the biomolecules attaching on top. Depending on how high the dopant concentration inside the wire is, there is different theories that can give you this depletion length. So if you have a very highly doped nanowire, above 10 to the 19th carriers per cubic centimeter, then the Thomas Fermi theory will give you the depletion length. This is indicated by this line over here. Whereas if you have a lower charge carrier density, then we can use the same theory as for the outside of the nanowire, the Debye-Hückel theory, and get the depletion length like this. Apart from this screening of the charges, which we don't really have much control over, we can further press the sensitivity of the nanowire, increase the sensitivity of the nanowire by using it at the correct regime. So here you have a nanowire that is gated by an electrode, and here you have the response, or the conductance of the nanowire as a function of gate voltage. Now, if we have the nanowire at a gate of -0.4, then we are situated in what is called the linear regime of the nanowire. Further away, at around 0, we are at an area where now the conductance starts changing more rapidly with gate voltage. This is called the near threshold regime. And there is a point here before we fully deplete the wire where we have the what is called sub-threshold regime. where we have almost fully depleted the nanowire, and this is where this slope is the highest. That means that if we're situated over here, any small change in gate voltage, for example, that is caused by bimolecules attaching on the nanowire, we have a large effect on the conductance. Indeed, if we change the pH in the solution at the three different regimes over here, we can see that in all cases there is a change in conductance that we can measure. But if we now plot the relative change in conductance, so the conductance, the sensitivity of the nanowire, the conductance divided by the base conductance, we can see that at the subthreshold regime, the change in conductance is actually the highest. There is a lot of applications for these systems. I chose two that I find very interesting. One is the detection of microRNA. So microRNA is basically RNA that is very, very short, 18 to 24 nucleotides long, and they are important for gene regulation, stem cell differentiation and development, and also are clinically important biomarkers for cancer. Now, in this particular paper, the authors have functionalized silicon nanowire with PNA. PNA is artificial DNA, so it's DNA basically that is not charged. When they then add a complementary strand of microRNA to this PNA, microRNA is negatively charged, so we get a change in conductance through the nanowire. And if we plot this response, we get a response over here with a fully complementary strand. If we add a non-complementary strand, then of course it doesn't bind. That means that there is nothing to change the conductance of the nanowire, so we get also a practically zero signal over here. But the interesting part is that it's so sensitive that even a one base pair mismatch will greatly reduce the signal from the fully complementary to just one single base mismatch. And of course, the signal is also concentration dependent and actually has a very good sensitivity. Another interesting application comes from using arrays of nanowires to detect, for example, the signals, the depolarization of the neuron axon and following it through the array of the nanowire. The last sensor I'm going to talk about is the impedance flow cytometry. And the motivation behind this system comes maybe from the need to be able to the last sensor we're going to talk about is the impedance flow cytometry. So let's look a bit at the motivation behind the sensor. So if you are in a production environment and you have bacteria there, then you have certain problems. For example, reduced product quality because of poor taste or unpleasant odors, the risk of having a reduced shell life for your product, and the worst case, if the bacteria is actually pathogenic, then there can be a serious risk of infection of both the production but also the product. This is currently addressed by regularly collecting samples from the environment, sending it to a laboratory where you grow them on plates, culture plates. Usually you have to wait 3 days for the result, and then you get the answer that yes, there were bacteria, or no, there were no bacteria. Now the way this is done, as I said, is on this growth plates, where you count the number of visible colonies. Now you need, for the different bacteria, you need to use different growth mediums, and the entire method is limited by whether or not the bacteria grows or not. Not all bacteria can grow, and not all bacteria grows on any medium. Ideally, you would like to not have to wait 3 days for the answer to see if you have bacteria in your production. The solution, or one solution, could be this impedance flow cytometer. This is based on the fact that you can take a swab sample, insert it in the device, press a button, and that counts basically in microfluidic channel the amount of bacteria in the sample. Because of the way that the method works, there is a certain concentration limit that you can measure in liquid samples. Samples could be water, swabs, or culture samples. It has a simple user interaction, and it's meant for basically getting the number of bacteria out. Now, this particular system that we are presenting here is developed by a Danish startup company called SBT Instruments that has actually sprung out of our research group. Let's look a bit at the bacteria. So bacteria are actually quite interesting organisms and with quite interesting membrane structures. So you have bacteria that is gram-positive or gram-negative. Gram-positive bacteria have a plasma membrane and then a thick peptidoglycan layer on top, whereas gram-negative bacteria have the membrane, a thinner peptidoglycan layer, but also an outer layer of a membrane that is rich on lipopolysaccharides. We are physicists, so for us it's not that important what is inside in the cytoplasm of the bacteria and how the membrane looks, but we can see the bacteria, the gram-positive ones, basically as a 3-shell model where we have the cytoplasm, the inner membrane, and then the peptidoglycan layer, whereas the gram-negative bacteria have an extra membrane, outer membrane. We usually use beads also for these systems, and beads in our very simple representation are basically a homogeneous polystyrene particle. Bacteria can be spherical or they can be rod-shaped, and the sizes of bacteria are sort of from 0.5 micron to 2-3 microns. Now, the principle behind impedance flow cytometry is that any particle coming between a set of microelectrodes that are generating an electric field in an electrolyte medium will disturb this electric field, and that disturbance of the field can be monitored. Now, usually we use 2 pairs of electrodes and measure the differential signal between the two. So we put a voltage on A and C, an alternating current voltage with usually 2 frequencies, and we measure the current in B and in D, and then we take the difference of the 2 currents. So if we have a particle traveling inside a channel like this, when the particle moves through and starts to changing the signal between A and B, then we will start seeing a change in the differential current, which is maximum when the particle is right in between the electrodes. Now, if the particle now comes out of this electric field, eventually it's in between the 2 pairs of electrodes. That means it now disturbs equally the 2 electric fields over here, and therefore we get back to a 0 signal. When the particle now moves through the other electrode, we get the exact opposite signal from the first one, and then when the particle has left the area of the electrodes, then we are back to our 0 signal. Therefore, any particle transition through the system will result into this kind of sinusoidal signal through, which is called the particle transition. This is a high-speed camera video of beads going through the channel with the two pairs of electrodes. So we're watching the electrodes from the top. So actually, this is the top and bottom electrodes, and the particles are coming through. And you can see that when the particles come through the electrodes, then we have a signal coming on. And if unfortunately there's two particles coming in at the same time, then we have some issues distinguishing that. Now, if we think a bit about the bacteria membrane and inside, we can see that well, the cytoplasm is actually a generally conductive area that has a finite conductivity and permittivity, and so is the periplasmic space. The membrane, on the other hand, is a very thin domain, about 10 nanometers, and it's insulating. So we can think of the cytoplasm as a resistor, conducting resistor, whereas the membrane can be thought of as a capacitor. If we now create a equivalent circuit model of our system, so we have our electrodes in the liquid. The liquid is an electrolyte, so it has a finite resistance and also a capacitance. And then we go to our particle, where we have the membrane, which can be thought of as a capacitance. But because there's also this proteins that the cell uses to communicate with the outside world, then we can also include a resistance. Then we have the cytoplasm, which is a conductor, and then again the other part of the membrane, and then back to our electrode. Now, a capacitor is a element that changes, where it has an impedance that changes with frequency. So at the low frequency, the impedance is high, whereas at the high frequency, the impedance is low. The resistor has no dependence on the frequency of the signal. It's not really that simple. We still have an electrolyte and we have a voltage in the electrolyte. That means we form a double layer. And that double layer should also be modeled to get the full equivalent circuit. And if we model the double layer as also a capacitor with the resistor in series, then we have the full equivalent circuit of the system, containing all the elements, including a parasitic capacitance, which comes from all the wiring of our chip. Now, if we for a second forget about that we have a particle in between the electrodes and just look at the electrolyte itself, then you can show that the frequency response of such a system contains an area that depends on the double layer capacitance, an area that depends on the parasitic capacitance, and then an area in the middle where there seems to be no frequency dependence, which means that we are now measuring through the resistance of the medium. That means that if we now limit ourselves in this area where we're measuring directly at the medium, so in what is in between the electrodes and not what is on the double layer or the parasitic capacitance, so if we limit ourselves within a frequency range, then we can ignore quite a few of these elements from the equivalent circuit and create a much simplified, more simplified circuit. Sometimes we can also ignore the medium capacitance. And then our cell model is actually extremely simple. Now, as I mentioned, we use a two-frequency signal, and if you remember, with the capacitance and the resistance, if we use a low-frequency signal, then the membrane capacitance will be basically insulating, so everything, the electric field lines will move around the particle. That means the low-frequency signal can give us information about the size and shape of the particle. At the high frequency, the membrane becomes transparent, and now we can probe the properties of the membrane and the inside of the bacteria. And when that happens depends on the membrane capacitance. That means that if we choose a high frequency here, then we can actually get a different response depending on the properties of the membrane. When we do these experiments, we usually get a huge number of particles through our electrodes, and then each particle is represented by a dot in this sort of plot, scatter plot. And there is a lot of things that we can plot. We get 4 signals out, a low frequency and a high frequency real part, and a low frequency and high frequency imaginary part, and we can plot any combination of this. Usually what we use is the opacity in cubic root scatter plot, where we have the size on the x-axis and then the high to low frequency ratio on the y-axis. Another scatter plot that we can use is the low frequency and high frequency argument. And this is actually quite interesting if you want to distinguish bacteria based on their viability. The reason for this is that in this sort of plot, you get particles that are effectively non-conducting in both the low frequency and the high frequency area signals on in this position 1. Particles that are conducting in both the low frequency and the high frequency signals are appearing in area 3. That can effectively be cells that have broken membranes. And everything else will be somewhere in between. Indeed, if we use polystyrene beads, they are insulating, and they appear here in position 1. Silver-coated silica beads that are conducting appear in position 2, and bacteria that have intact membranes appear in mostly in position 2. Now we can use this fact by studying viability of bacteria. So the problem with bacteria viability studies is that if we just use growth of bacteria, then we run into the problem that not all bacteria can be cultured. However, we can still see them passing through a set of microelectrodes. That means that with impedance flow cytometry, we can actually detect viable but non-culturable cells. So we have the possibility of detecting bacteria that conventional methods cannot detect. On the other hand, biological viability is huge, so we can actually have false positive signals. Now, we wanted to see how IFC impedance flow cytometry measures viability of bacteria. So we used a strain of E. coli that is culturable, and we inactivated the strain by 3 methods, either by adding ethanol to the bacteria, heating it at 90 degrees, or doing something radical, autoclaving it at 125 degrees. For each of these inactivation methods, we checked the viability of bacteria using 3 methods: plate counts, where we simply, after inactivation, try to group the bacteria in the proper plates. We also did a live dead assay with fluorescent staining. So bacteria that is alive appears green, whereas bacteria that have perforated membranes appear red. And then we also used the impedance flow cytometry method and to try to see what we could get out of it. Now, what we expect is that at the low frequency signals, both the beads and the untreated bacteria will be bypassed by the current because of the membrane capacitance. But when the bacteria is inactivated, then because we create holes through the membrane, then we will be able to probe the inside of the cell. At the high frequency, we expect to get both the untreated and the inactivated bacteria to bypass the membrane, which means that we could see a difference between properties of the fully functional membrane and the perforated membrane. Now, indeed, after we inactivated with all three methods, we could not see any growth on the plates, and the fluorescent images show broken membranes in all inactivated samples. However, there is a difference in the impedance in all the inactivated samples. The beads, they appear here in position 1. Note that this graph is inverted, irrelevant to what I showed you before. In the ethanol-treated bacteria, the bacteria actually shows up where we expect them, in position 3, broken membranes, relative to the green dots over here that are the untreated bacteria. However, in both the autoclaving experiments and the heat experiments, we see something completely different. If we now plot the high-frequency argument and create a log plot of this high-frequency argument and use this as a viability indicator, we can see indeed that there is a very good response for the ethanol and a very decent response for the autoclaving, depending on the threshold that we use. However, the heat basically shows almost no distinction between viable and non-viable cells. But all of these bacteria were indeed inactivated, as the other methods indicated they were not growing. Now, to investigate this further, we increased the heating time from 5 to 20 minutes. And indeed, we see that the untreated bacteria is showing this banana shape as we know it, but every time we increase the time of heating, then we slowly move the bacteria to position 3, like with the ethanol. And this can also be seen in the log plots where we have the curves moving towards the corner here, where we have the best the best differentiation between viable and non-viable bacteria.